am ready.
Okay, so thank you everybody for joining us at our very first virtual Marine Science Day. My name is Cinnamon Moffett and I'm the Research Program Manager at Hatfield Marine Science Center located in Newport, Oregon. And I'm your host for today's main stage talks at Marine Science Day. You might have already figured it out, but this is a webinar format, which means your camera, screen shares, and mics have been turned off, but we do hope that you chat with us, as many of you have been doing in the chat box. So you can find that either at the bottom or the top of your screen. You click the little box above chat and you'll get a little pop-up and you can put your questions there. Feel free to do that at any time, and then Lisa and I will work through them at the end of her presentation. I also wanted to let folks know that this talk is being recorded and will be posted on the Marine Science Day web page in a few days. So if you missed something, you'd like to see it again, or you'd like to share it, feel free to come back to us, join Marine Science Day's page again in the future. It will be up and live for a year, so go ahead and come back and visit us again. I also wanted to let you know that this is a series of talks on the live stage. So we have one more after Lisa. The next one will start at 1:35 and is Taylor Chappell, and he'll be talking about sharks along the Oregon coast. So come back for us there. And then what you're all here for is to hear Lisa talk. And so Lisa's going to be talking to us about, do gray whales count calories? But before we get started, just a little quick background on Lisa. Lisa is a third-year graduate student at Oregon State University in the Department of Fisheries and Wildlife under the supervision of Dr. Lee Torres in the Geospatial Ecology and Marine Megafauna Lab, more commonly known as the GEM Lab. And so I'm going to hand it off to Lisa. Lisa, the floor is yours.
Awesome. Thanks so much, Cinnamon. Hi, everyone. Hope you're enjoying your virtual Marine Science Day. Yeah, like Cinnamon said, So currently a PhD student. I defended my master's in the GEM Lab with Lee in December, and that's what my talk today is going to be all about. Talking about whether gray whales count calories. So before we can dive into that question, let's talk a little about gray whales first. There are currently two populations that exist of gray whales. There's the Western North Pacific population, which is quite small, about 150 individuals, and then the much larger Eastern North Pacific population. Historically, there was also a third population in the North Atlantic. However, due to historic whaling, this population unfortunately no longer exists. So today we're interested in the Eastern North Pacific gray whales, which migrate annually from their breeding grounds in Baja California, Mexico, to their feeding grounds in the Bering, Chukchi, and Beaufort Seas up in the Arctic, where they predominantly feed on benthic amphipods. So unlike other big baleen whales that will filter mouthfuls of water to obtain their prey, these ENP gray whales suck up mud and benthic sediments to filter out these amphipods. And this population is estimated at about 25,000 individuals, but they have experienced two unusual mortality events in the last 2 years. And an unusual mortality event is declared by NOAA when the stranding rates in a given year are above the average stranding rates that we typically see for that marine mammal population. And there's a couple of theories as to why this has occurred twice in just the last 2 decades. One is that the population may have reached carrying capacity, which means that the population has kind of grown to a point where the prey can no longer sustain that number of whales. And that's kind of tied to the second hypothesis, which is that environmental shifts in the Arctic due to climate change has changed kind of that prey base, how much is available and what's there. And that's limiting the population and causing these UMEs. There is a little subgroup of the ENP called the Pacific Coast Feeding Group, which contains about 250 individuals. And they cut their migration short, they don't go to the Arctic, and instead they spend their summer foraging seasons along the Pacific Northwest coastline from Northern California to Southern British Columbia. And in this range, they occupy really shallow nearshore habitats where maybe many of you have seen gray whales from shore before here in Oregon in the summer. And in this range, they have a really broad diet. So they don't just feed on these benthic amphipods, but they feed on a number of different species. And this is where we're faced with a puzzle, kind of. We have to ask, why does the Pcfg exist? Well, from the map, we can see that the Pcfg migration is about half the distance of that of ENP whales. So, I mean, why not feed here instead? Why not come to the Pcfg range? But why then is the PCFG group so small compared to this larger ENP population? Why don't more whales do this? It's especially puzzling that the PCFG is such a small group, since work done in the GEM lab by my former lab mate, Dr. Leila Lemos, has proven that PCFG gray whales successfully gain weight during the summer feeding season along the Oregon coast. So what Leila did is that she took images from drone footage, as you can see here on the left, to compare body condition of PCFG gray whales and how it changed over the course of the foraging season over 3 different years. So this plot on the left shows that at the beginning of the foraging summer season, PCFG gray whales are really skinny, but by the end of it, they're at their fattest. So showing that they're able to progressively gain weight on the feeding grounds here. And this weight gain is really important because whales have to gain all of the mass that they're going to need for the subsequent year for migration and possibly reproduction in just those 5 to 6 short summer feeding months. Another reason why it's surprising that the PCFG is so small is that we know that female gray whales that repeatedly use the PCFG range as their summer feeding season have successfully reproduced and supported calves. So take this famous gray whale up here. She's known by many as Scarback, although we like to call her Scarlet because we think Scarback is a little too ugly a name for such a resilient, amazing whale. As you can see in this photo, Scarlet has sustained a massive injury to her back many years ago, which has left her with this permanent concave wound. But despite this, Scarlet has persevered, and in just the 5 years that the GEM lab has been conducting research on the Oregon coast, we've seen her with 2 calves, one of which is pictured here. So what this photo is this head belongs to Scarlet, and this head belongs to her calf Rose, who we saw last summer. So given that gray whales are able to gain critical mass on the PCFG foraging grounds and females successfully reproduce and support calves, this is where we're met with this puzzle that has these kind of 2 questions at the center. Why does the PCFG exist, and why is the PCFG so small? Migrating to the PCFG range is shorter. It's a shorter distance, so it's probably one of the reasons why the PCFG exists in the first place. However, there may be increased foraging costs associated with feeding in the PCFG range, and therefore, this could be why the group is so small. It may also have something to do with differences in prey between the Arctic and the PCFG, differences that could relate to quantity, quality, or availability that makes it more worth it for whales to migrate all the way to the Arctic. As I mentioned before, it could also be environmentally driven, that conditions always fluctuate in the marine environment, and so that can kind of drive where whales decide to go every summer. It could also be culturally transmitted, that certain females that come to the PCFG range will pass that behavior and that culture onto their calves. Or we could also consider that there might be increased risk for whales that forage in the PCFG range, since their nearshore habitats overlap heavily with a lot of human activities, including vessel traffic, whale watching, and fisheries, to name a few. And some of those aren't as present or as common up in the Arctic, which is more remote. So for my master's work, I wanted to look at prey quality. I wanted to look at this question, whether prey quality is the driver of these 2 different population sizes. And I hypothesized that prey quality must be better in the Arctic, and that's why the ENP population is so much larger than the PCFG. So to get at that question, I first had to collect a bunch of Oregon prey samples. And the way we did this is by using a light trap, which you can see on the left here, which is basically just a water jug with 2 attached floats, and it's weighted down with a cinder block, which you can see better here. So we deploy it over our rib, and the anchor keeps the light trap in place, but the floats allow the trap to sit about a meter above the sea floor. And that's because we're hoping to capture epibenthic prey, which is very common in our nearshore Oregon reefs. And inside this light trap, you can see this teeny tiny light. We put that in there and that attracts the zooplankton overnight. And then the next day we can collect the light trap and hopefully have a bunch of zooplankton in there. So we did 36 light trap deployments over 3 years, which you can see are these circles on the map. And we were also able to collect 3 opportunistic surface net samples, which are the triangles on this map, which were of 2 different species of crab larvae, which we actively saw gray whales feeding on. Once we had these samples, we sorted them to species level and into reproductive stages. So in order to find out what the caloric content of a prey item is, and the caloric content basically is just the amount of energy in a prey item, you have to use a method called bomb calorimetry, which sounds threatening. It isn't to humans, though it is to the little zooplankton. But the way it works is that you dry out your samples in an oven. I then ground all these samples into a powder, form them into a pellet. I then had to build what is called the bomb, and that is the official term in the manual, not something that I made up. And the little pellet gets placed inside the bomb. You screw it all together, it gets filled with oxygen, and you put in the calorimeter, and that it will combust the sample, and the calorimeter measures the temperature difference, and then magically it outputs a caloric value for me to process and analyze. So what we found in our light traps and from our surface samples were 6 predominant prey species on the Oregon coast. We had these 2 different types of amphipods, 2 different species of mysid shrimp, and we collected 2 different kinds of crab larvae. And we count these 6 species that we found in Oregon. We use them as a proxy for prey in the entire Pcfg range because of the similarity of prey species and habitat types that have been described throughout the range from Northern California to Southern British Columbia. And then for our Arctic comparison, we got published values from the literature for this one amphipod species because it comprises over 70% of the benthic amphipod community in the Bering and Chukchi Seas, and it is the documented primary prey of ENP gray whales. So first up, what did we find in Oregon? So what you're seeing here on the horizontal axis are the 6 prey species that we collected that I just described, and on the y-axis we have their caloric content. And what we found was that yes, caloric content does vary between species, and this was mostly driven by the fact that these 2 species, Neomysis rayi, which is one of our mysid shrimps, and Dungeness crab larvae had significantly higher caloric contents than all other species that I tested. And when I then compared those values from Oregon prey as a proxy for Pcfg prey to the Arctic benthic amphipods, I was quite surprised with what I found, because the 2 species in my study that had the highest caloric contents also had higher caloric contents than the predominant amphipod prey in the Arctic. And this was quite shocking because if you can remember the puzzle piece I was investigating for this study, I had hypothesized that prey quality in the Pcfg range must be lower than the prey quality in the Arctic in order to explain the Pcfg size and it being so much smaller than the ENP. So to look at this a little further, I extrapolated these prey caloric values to daily gross energetic requirements of gray whales, which I obtained from a fellow graduate student up at UBC. And so what you see here on these plots is on the x-axis, we have age and years of gray whales split out by 3 reproductive stages. We have lactating females on the left, pregnant females in the center, and on the right, we have all others. So that's non-reproductive females and males. And on the y-axis, we have the metric tons of prey per day that a gray whale would need to consume in order to meet their daily energetic requirements. And the different colored lines that you see represent the different prey species. To make this a little easier to understand, because there's a lot going on, I'm just going to highlight the 3 most interesting prey species here. We have Dungeness crab larvae in orange, Neomysis rayi, that mysid shrimp, in pink, and in black we have the Arctic benthic amphipod. So if we consider a 30-year-old lactating whale and what she needs to eat every day in order to hit her daily energetic requirements, if she could eat Dungeness crab larvae all summer long, that whale would need less than half the amount of food than a whale that fed on Arctic benthic amphipods. Therefore, the foraging energetic cost of capture for a whale feeding on the Arctic amphipods would likely be twice as high. So then why? If Dungeness crab larvae are such caloric gold mines, don't more whales spend their summers in the Pcfg range to feed here? And this is where I tell you that this plot is a little misleading, and that's because Dungeness crab larvae aren't available the entire summer in the Pcfg range. And that's because of their pulse reproduction. So we tend to see kind of swarms of crab larvae in mid to late spring, sometimes into early summer. And so for this reason, a whale can't actually feed on crab larvae all summer long. Nevertheless, though, if you look at this pink line, a whale would still need to eat fewer of these mysid shrimp than Arctic amphipods. So then why do we see this trend when clearly there are prey species available in the Pcfg range that are more energetically rich than Arctic prey? Why do we have this mismatch in prey quality and gray whale population sizes? And the answer is we don't really know at this point. But what my study has shown is that prey quality is not higher in the Arctic than in the Pcfg range. So prey quality probably isn't the driver of the difference in these population sizes. What I hypothesize is that it's probably a question of prey availability and prey quantity. I think prey is more consistently available and in larger quantities in the Arctic than in the Pcfg range. And coupled with this, foraging on these diverse prey species in the Pcfg range may cost more energetically for the gray whales than simply feeding benthically on amphipods, because these gray whales have evolved and developed this benthic feeding strategy for over centuries now, so they're probably really efficient at it. So while I don't have a definitive answer here yet, and there's still a lot of question marks in this puzzle, what we do know is that Pcfg prey have significant differences in their calories. And because of this knowledge, I wanted to go further to see whether the differences in prey calories affects Pcfg gray whale foraging. So to do this, I examined the foraging decisions of Pcfg gray whales relative to prey quantity in terms of abundance and prey quality in terms of caloric content. In order to do this, we had to collect prey data and whale data at the same time and location, which is tricky because baleen whales tend to, forage in the ocean, and they're hard to keep track of because they dive and they disappear. But luckily, since gray whales on the Oregon coast forage so close to shore, we found a way to do this in Port Orford along the southern Oregon coast. So there we undertook prey assessments from a research kayak, where we collected prey samples with a net to assess prey quality by looking at what prey was there and then tying that to the caloric value analysis that I just done. And we used a GoPro camera to assess the prey quantity to know how much prey was in an area. And at the same time, we had a team on shore tracking whale movement and behavior. So what does this all look like? What you see here is a map of our study site with this gray area indicating the shore. And this black circle indicates the cliff site, which you can see up here. So up on the cliff site is where we had a team of people who tracked whales as they came into our study area in Port Orford. At the same time, a research kayak would launch from this beach here and go to two different study sites, Mill Rocks and Titchener Cove. And the kayak would conduct prey sampling at each of these blue numbers. So those blue numbers are kayak sampling stations where we've been repeatedly sampling the prey for over 6 years now. So how do we track whales from shore? It sounds a little counterintuitive. Well, we use this thing called a theodolite, which you see right here. And a theodolite is a surveyor's tool that uses a known height, location, and angles to calculate a precise GPS location for whatever it is you're pointing the theodolite at. So in our case, gray whales. And so every time the gray whale comes to the surface, you can fix its location with the theodolite and get a very specific GPS location. And so this track line is a very basic map of what the theodolite produces. All of these little yellow dots that you see is a location where a whale came to the surface, and we fixed its location with the theodolite. And those dots are connected with one continuous line. And so for the purpose of my study and the questions that I was trying to ask, I needed to know what behavior state a whale was at each of these yellow points. I wanted to know whether a whale was foraging or searching or traveling. So how do we assign behavior states to just points in a track line? Well, we use this method called residence in space and time, which assigns behavior state based on occupancy patterns in space and time within a given radius. So following this method, a whale that is transiting or traveling through an area spends very little time in a radius because it's just moving through quickly and trying to get somewhere else. A searching whale, on the other hand, will spend a lot of time and distance within a radius as it's moving through an area looking for prey to consume. Once a whale has found prey that it wants to consume and begins foraging in an area, it'll spend a lot of time within a given radius, but it won't cover a lot of distance. And that's because the whale wants to make sure that it gets all the yummy mouthfuls of prey that it's just spent time and energy searching for. So now that I had all my whale data, how do we collect prey data from a kayak? To best show you how we do this, I'm going to show you a video of one of my former interns, Haley Kent. And so I hope the video comes through okay and isn't too choppy. So first of all, Haley is dropping a Secchi disc, which we use to measure water clarity. Next, she's going to drop a GoPro stick, which is just a GoPro with a time depth recorder that we use to assess relative prey abundance. And finally, our zooplankton net is used to collect a representative sample of prey at that sampling location. So how do we get relative prey abundance from a GoPro video? Well, what we do is we extract screenshots every 5 seconds from the upcast of the GoPro, since we do this at a consistent speed. These screenshots are then gridded up in a 3x3 cell grid, and each grid cell is assigned a relative score from 0 to 5. 0 meaning there's no zooplankton, score of 5 meaning that it's chock-a-block full with zooplankton. So as an example, here I've scored these boxes. I then took an average value of all of those. And once that was done for all screenshots of one GoPro cast, I summed all of those together to get a relative prey abundance. Then from our net samples, we would sort every single one of these teeny tiny critters and zooplankton that you see. We sorted them to species level. And from this, we found 3 predominant species accounted for 95% of all individual prey items. And these 3 names, or maybe these 3 images, may look a little familiar to you because it's one of the amphipod and both of the mysid shrimp species from my caloric assessment. And so if you've forgotten those results, that's completely okay. But the one thing to remember is that Neomysis rayi has a significantly higher caloric value than the other 2 prey species. So once we sorted all of these net samples, we then calculated proportions of each species per sample per day. So once I had the quality data and the quantity data, I was able to integrate both of these together to create these caloric abundance spatial layers to know what kind of prey was available to whales on any day that we sampled for prey. Then we can bring in the whale data, where here you see a track line of a whale called Smudge, who we saw in Port Orford a lot in 2019 and 2020, foraging quite heavily in one of our study sites. As you can see, there's a lot of points there and they're all in a really tight cluster. So since we sampled for prey on the same day, we can overlap the whale track onto the prey layer for that day and see what areas Smudge foraged in more than others, and whether that was driven due to prey quality or prey quantity, or kind of a trade-off of both of those things. So I did this for every sampling day and whale track line that we had. And so the results that I'm going to show now is kind of a summary of 3 years of data of both whales and prey. So what you see here is called a violin plot, and it shows the distribution of prey values at whale locations by the 3 different behavioral states: forage, search, and transit. And the little diamonds show the median or the average value. So first of all, what we see is that whales foraged more in Neomysis rayi areas than they searched and they transited in. However, the same trend is not evident for Holmesiomysis sculpta, our other mysid shrimp species, where whales searched more than they foraged or transited in. Secondly, whales never foraged in areas where the amphipod Atylus tridens dominated the prey community, even though whales searched and transited through these areas, suggesting that whales actively avoided this species. And thirdly, as you can see, the median abundances of Neomysis rayi are lower than those of the other 2 prey species, indicating that this species was less available throughout the study area. And all of these taken together were interesting to us because it hints at a preference for some prey species over others. And so to get at that question further, we apply generalized linear models to answer these questions. So what you see here is the number of whale points within a track line, color-coded by behavior state once again, against the mean available relative abundance of prey. And so what this analysis revealed is that when prey species were grouped together, so just cumulatively all prey that was in an area available to a whale, whales significantly increased their foraging effort as prey availability increased. But there was no change in their search or their transiting behavior with increased prey availability. But when looking at this split out by species for our 2 mysid shrimp, we see some interesting results. For Neomysis rayi, which remember has significantly higher calories than the other 2 prey species, whales significantly increased their foraging behavior as abundance of Neomysis raei increased, while significantly decreasing their searching and their transiting time. In contrast, though, for Holmesomyces sculpta, the only change in whale behavior is that whales significantly increased their searching behavior when sculpta abundance increased. And this suggests that whales are never quite satisfied when they encounter this prey species, as they continue to search rather than switch to foraging. And this was surprising to us because surely if prey abundance increases, so there's just more prey available, a whale should increase its foraging behavior. But since we know that Neomysis raei is a calorically richer prey species than Holmesomyces sculpta, what this meant to us is that whales do, perhaps at this micro scale, prioritize prey quality over prey quantity when making foraging decisions. And so what this means is that Pcfg whales do display selection and avoidance of certain prey species based on their caloric values. Because remember, whales never foraged in areas dominated by the amphipod Attylus tridens, which had the lowest caloric value of the 3 prey species in this study, but they always selected to forage in areas dominated by Neomysis raei. Therefore, prey quality impacts the microscale decisions made by whales while foraging, and prey quality shouldn't be overlooked in studies of predator foraging ecology. And so what this all just means is that understanding the prey is really important in allowing us to understand the predator, which is why I've been working a lot on prey for the last 2.5 years to understand gray whales. I think often people are kind of confused when I spent so much time in the lab and sorting these little species, but it's really important to understand the prey base, to understand the dynamics of our predators and how best to manage and conserve them. And so with that, I have a lot of people to thank. Primarily, it's all the interns that you see on the left here, and thanks to all my funders. And I'll also just say thank you to anyone who has an Oregon whale license plate, because those funds helped support this Port Orford project. And so with that, I will take any questions that there may be, and thanks for having me.
Great. Thank you so much, Lisa. We've had a very active chat, and so I will try to pick out the questions out of the chat, so be patient with me.
Okay.
Can you talk about prey availability in the 2 different foraging areas, the Arctic and the Pcfg, and whether the prey is available at the same time at the same, like how those 2 compare temporally?
That's a really good question. So it's a little hard to assess prey available. So far it's been really hard to assess prey availability here in the Pcfg range, because these predominantly mysid shrimp are in these really kelpy, rocky, shallow habitats, and it's difficult. You can't really do your traditional tows because of potential entanglement of the nets in the kelp. We've tried that before and have always had entanglements happen. So it's quite tricky to do that. And then also trying to do it with acoustics can also be difficult, because it's such it's so shallow and there's so much kind of background noise from the reef and the kelp. So it's been tricky to try and assess the availability of prey in the Pcfg. A little more straightforward in the Arctic, but it hasn't been done for a really long time. So actually a lot of the studies that I'm going off of about prey availability are from the 80s. And then there was recent assessments kind of in the early 2000s, which did show that prey availability has declined in the Arctic in those kind of 20 years. But the prey is available at the same times. I mean, that's why the whales go to forage in the summer.
Great. Can you talk a little bit about how you compare the GoPro prey availability? I will start again. Compare the GoPro relative prey abundance versus the sampling in the nets.
Oh, good question. So I kind of breezed over that, but we sample both of those at the same location. So there is a bit of a delay where we drop the GoPro first and we then do our net sample, but we kind of have to, for science's sake, assume that we're hitting the same location there and that we're able to compare kind of how much prey versus what species.
So there are so many more questions than we can get to in the 1 remaining minute. So what I want to do is, for everybody who put in questions, thank you so much for engaging with us. We really appreciate it. If you would like to follow up with Lisa about your questions because we weren't able to get to them here today. Her contact information there is on the screen, and you're welcome to reach out to her directly with those questions. The other thing that's happening, Lisa, is yay and thank you and such a good job and you're awesome and standing ovation and all those great things. So virtual thanks from everybody in the audience. Lisa has just put her direct contact email into the chat so you can reach her there. And again, I apologize for not getting to all of the questions, but thank you so much. For those folks that are online, just wanted to remind you that Taylor Chappell will be speaking with us about the sharks in Oregon coast. Starting at 1:35. And again, Lisa, thank you so very, very much for joining us today. For everybody else, you get a few minutes to go explore Virtual Marine Science Day. Check out the GEM Lab virtual exhibit in the exhibit hall. Go to the Kids Zone and apparently check out the art wall. It is growing and is apparently looking amazing. So go check it out and then come back and join us at 1:35. Thanks, everybody.
Yeah, send me emails if you have questions. My email's in the chat.
Perfect. Thank you for working word edgewise for Cinnamon. All right. Thank you so much, everybody. And thank you, Lisa. We are going